Everyone. So this will start our sixth effort in this series of this collaborative interactive learning group on whole genome sequencing. So this is presented to you by the New York State Integrated Food Safety Center of Excellence. And our goal is really to make this interactive. So I will ask questions, I will look for feedback. If you have a camera and try to remain anonymous, I understand that, but I would really appreciate if some of you at least turn on the cameras, because it really does facilitate more sort of interactive efforts and discussions here. So what we're going to do today in this series is to talk about whole genome sequencing, how it is used to analyze pathogen isolates from foods. In the previous parts of this series, we already talked about whole genome sequencing human isolates. How do epidemiologists use these data to detect, trace back foodborne disease outbreaks? Today, we're really going to start talking about, investigations and efforts that start from whole genome sequencing-based characterization of food isolates. So the learning objectives are to sort of continue to emphasize how whole genome sequencing data are used in epidemiological investigations, in foodborne disease outbreak investigations. And that's really sort of deepening our understanding from previous efforts in the same series. But then the very specific learning objective for this one is to understand how whole genome sequence data are used in routine characterization of pathogen isolates from foods and food-associated environments. And food-associated environments may mean that a FDA, USDA, or a state ag and markets department goes into a facility, collects an environmental sponge and characterize that. What I'm going to cover is sort of a quick overview review of whole genome sequencing-based subtyping. Some of you may or may not have been at the previous installations of this, and then focus a little bit on background and pathogen ecology and processing plants. What do we know about foodborne pathogen and processing plants and food-associated environments, and from that then move into some of these more interactive case studies. And if you have questions really at any time, you can type a question in. There's a way to do this on the chat, or you can raise your hand, or you can just interrupt me and ask a question. That's really my hope, is that this is interactive, and if something doesn't make sense or someone has to add something, you will add it at any time. So let's start with a short review of whole genome sequencing-based subtyping. What I really want to focus on here is not the actual sort of technology on how you take a bacterial isolate and determine its whole genome sequence, which is, depending on what pathogen we talk about, between 3 and 6 million ACTs and Gs, but really talk about the data analysis. Today, we really have two, some people may call it competing analysis approaches. But these are not really competing, they're complementary. The two approaches are high-quality SNP, so HQ SNP analysis. And this is what's predominantly used by FDA in the US and some state health departments. And then the other approach is a whole genome sequencing MLST approach. This is the approach that CDC is spearheading. This is at least where we are right now, where whole genome sequencing and the way whole genome sequence data are going to be analyzed and communicated through as we replace PFGE with whole genome sequencing. The important take-home messages from here: both of these analyses are conducted in the same set of raw data. When you sequence a whole genome of a Salmonella listeria, you can run either method. You ran one, you can go back whenever you want to and run the other method of analysis. Both analyses typically give you the same or very similar conclusions. They use different words, but the conclusions rarely are different. Now, that doesn't mean they can't be different. They might not sometimes be different. Typically, they give you the same conclusions. The differences are that high-quality SNP analyzes the whole genome assembly. And whole is sort of maybe an exaggeration, about 90, 95% of the whole genome, for relatedness. And this relatedness is expressed as SNP differences, single nucleotide polymorphism differences. This one is a bioinformatically somewhat more challenging approach. It's not amenable to an easy nomenclature where you say this is subtype A and this is subtype B, but it is good for a situation where we don't have the whole genome MLST database. It sometimes might provide better discrimination than whole genome MLST, and often is used and can be used in conjunction with whole genome MLST to just confirm that both approaches give the same conclusion. Whole genome MLST, on the other hand, analyzes data gene by gene. It's easier to perform, and it provides you with an easy nomenclature that some people call the whole genome MLST addresses or zip codes, and I'm going to show you some examples of that. Just to illustrate, here's your typical SNP. On the top, you see what we typically this refers to. One isolate might have a C, another one has a G at a certain location. That would be a 1 SNP difference. And we look at really these SNPs again over 3 to 5, 6 million nucleotides. If we have data on a large set of isolates, we typically express them as trees. And then in these trees, we can do some cute color coding and other things. And in this one, you can see, if you look at the very bottom, you have 4 isolates or 5 isolates that are color-coded in yellow, and they all differ by 0 to 3 SNPs from each other. And so that says, okay, there's something going on. We got 5 isolates that are very similar. One of them is an environmental isolate, 4 of them are humans. So that gives us some idea about a potential outbreak and maybe a potential source. On the other hand, if we talk about the whole genome MLST, here we don't look at all 3 to 6 million at once, but we take these data and parse it out into smaller subunits. And the subunits are called loci, obviously singular is locus. And we can think of these as really different genes. So it's 5 million nucleotide genome is 5,000 genes. So rather than analyzing all 5 million at once, we analyze it gene by gene, or locus by locus. And we can look at gene 1 in one isolate here. So this is the sequence. We can look at a second isolate saying, well, this gene is different. Got a couple differences here. So we call it type 2. We look at a third isolate, say it's different, we call it type 3. How do you combine that to come up with a final subtype? Hypothetical 3 isolates, A, B, C. Hypothetical large number of loci or genes, 1 through 2005. Look at the first locus, all, let's say, 1,000 nucleotides of it. All 3 isolates are the same. Locus 2, isolate A and B are the same. Isolate C is different. Locus 3, isolate A and B are the same. Isolate C is different. We then look at all of them. If 2 isolates are the same at all loci, we call them the same MLST type, type A. If they're different, we call them B. We can quantify the number of differences as allele differences. So in this case, if we only had these 4 genes, C and B would differ in 2 alleles. Loci, locus 2, locus 3, the same here and here. So here our nomenclature now becomes allelic differences, not SNP differences. The problem with that is if you have 2 isolates that are different, have a different name, as shown in this previous slide, A versus B, we don't know how different they are. Are they different by 1 allele or are they different by 500 alleles? This has been resolved by an approach where we, rather than giving it 1 number, we give these MLST or these whole genome MLST types a code that typically it's probably going to consist of 6 different numbers. If the numbers of that zip code is the same, like in the first example, patient 1 and 2, that means these 2 isolates from these 2 patients are exactly the same. There's not a single allele different. If we have a zip code that differs, but it just differs in the last number, as shown for patient 3 here, then this one differs from patient 1 or 2 by anywhere from 1 to 7 alleles. So that means they're pretty similar. They typically probably could be considered part of the same outbreak. If isolates differ even more, 8 to 15 alleles, now the next number in also becomes different. So it's very similar to, a classical zip code. If 2 zip codes differ by the last number, you know that these 2 places are pretty close to each other. If they differ by the last 3 numbers, you know these 2 locations are very far away. So a lot more intuitive, a lot easier for nomenclature, and it tells you same or different, but it also tells you if they're different or they're in this proximity to each other, where you might want to investigate them as part of an outbreak or not. And you can then build trees very similar to what you can do with high-quality SNPs. Here you can see these 2 isolates have exactly the same zip code, so they're right next to each other on the tree. Here's one that differs just by the last number, and therefore it's very similar, and it forms a cluster here. So you can see that these zip codes have the potential to be very easy to interpret, and that is one of the reasons why CDC is going with this whole genome MLST approach and this idea of giving subtypes zip codes like this. Of course, even with these ones, zip codes, it's even easier to report these data. So you can simply, a lab made to an epidemiologist, just provide reports like this. Hi everyone, we got a new cluster. Here's our cluster name. That includes 2 isolates, let's say 2 Salmonella isolates that are exactly the same. Or we can say, we got a new cluster, we have 2 isolates that are 2 alleles different from each other, and typically they're probably also going to give you the isolate zip code. So this is the type of information you will get. So that covers our sort of background, a little bit of a repeat, refresher on whole genome sequencing, different analysis methods, whole genome MLST, high quality SNP. Going forward, 2018, or as Listeria monocytogenes becomes more rolled out and data analyzed by CDC, those are the types of data that you should expect to see. Zip codes, whole genome MLST data. Now let's move into the more specific part of today's session, which really focuses on whole genome sequences of isolates from foods and food processing plants. There are a couple background slides that really set us up for this. So the first one just illustrates what happens with regard to pathogens in food processing plants. And there I'm using Listeria as a model system. So this gives you some subtype data from the pre-whole genome sequencing era, where we visited one processing facility 5 times. We collected samples from raw materials, and the environment. You can see some of them are floor drains. Food that was in the process of being processed, so it's in a salt solution as it's prepared for smoking, etc., and finished product. Every time we found Listeria monocytogenes, we fingerprinted it with a banding patterns, as you can see here. This is ribotyping, but could be PFGE. And the interesting thing we found is that every visit to this processing facility, we found the same subtype of Listeria monocytogenes. It's this pattern here, and you can see it's named 1039C and marked by a star. We find the same Listeria on fish where the raw material came from Alaska, Droll Red King, where the raw material came from the Faroe Islands, where the raw material came from Norway, and we find it in the processing plant environment. Most likely explanation, that Listeria just lives in that plant over time, because between visit 1 and 2 was about 5-6 weeks, visit 3 was another 5-6 weeks later. Now, the real cool thing in this study was that we looked at 3 smoked seafood plants, all within 15 miles of each other, all producing very similar product, often using similar supply chains. Plant D that I showed you, the subtype we call 1039C, we found in 10% of the samples we collected in the facility, 23 times. Never found it in plant C, never found it in plant B. We go into plant C, this subtype here, we found multiple types here, never found it in B, never found it in D. So suggesting that these plants are colonized with very specific Listeria that survive over time. Some plants are colonized by one Listeria, some can be colonized by multiple Listeria. Plant B, about 10 miles from plant C, 15 miles from plant D. This subtype here, 1042C, found it every time we visited the facility. 1045, found it every time we visited the facility. But 1042C, we found once in plant C and once in plant D over the whole study. 1045, we never found in plant C, but a couple times in plant D. So colonizes this one plant, but something with a similar fingerprint may show up in another facility every now and then. But that brings up that idea that these bacterial, particular listeria, can survive in a facility over time. Another example, processing facility, we sampled raw material. We sampled the raw material area where raw fish comes in. We sampled the area where the product is produced. We sampled food contact surfaces, so these are slices and things that touch the food right before it's packaged. And we sampled the finished product. Whenever we found Listeria monocytogenes, we fingerprinted it. With barcodes, different Listeria were assigned different colors. Every column is a month, so we sampled this facility over 12 months. What you can see here is in the raw area, we pick up this Listeria that's color-coded in green every now and then, about half the times. We also pick it up once in the finished product area, but it seems to be predominantly residing in the raw material area. But there's no one spot where we pick it up every time we sample. So we did some more sampling, picked some more location. One spot, we find this Listeria every time we sample it. That seems to be a spot where that Listeria survives over time in that facility. This was another year of sampling. We found this same Listeria subtype in this plant over 2 years in the raw area, but it occasionally would move into the finished product area. And once we found it on food contact surfaces, including a slicer, which probably means it makes its way occasionally in the finished product. But the key is this same Listeria found in this facility over 2 years. Where did we find it? In a floor mat. The floor mat was about 1.5 inches thick, was sanitized by either spraying sanitizer foam on it, submerging it in sanitizer, but wasn't enough for that sanitizer to penetrate through and through. So the Listeria found a spot inside where it was protected from sanitizer. You put a 150, 200-pound person on these floor mats in the morning. With that weight, that Listeria gets squished back out, recontaminates the facility. We removed the floor mats from this facility. In our 6 months follow-up, never found that same Listeria again in that specific facility. This is just one example of what we call sort of niches or hiding places. Another classic is rollers that help conveyor belts. So you see a roller here, you see a conveyor belt, you see a little crack. That roller is hollow. Dirt can come in, Listeria can come in and distribute all throughout that roller. Sanitizer doesn't get in there very efficiently, if at all. So Listeria in the roller is protected from sanitizer, can grow and multiply. That roller rolls for 8 hours during production. You have some moisture seeps in, moisture seeps out, Listeria comes out, contaminates the conveyor belt, and if there's food in the conveyor belt, contaminates the food. Another perfect bacterial hiding place. And you find these same hiding places in retail establishments. We've seen them in deli cases. You can find the same type of hiding places in restaurants, in hospital kitchens, wherever you go. Places where these pathogens can survive over time. How extreme does it get? This is an example of a listeriosis case, 1988. 12 years later, 2000, same processing plant responsible for an outbreak with 29 cases. Same Listeria subtype from 88 and 2000. When we did whole genome sequencing on Listeria from 88 and 2000, we found that these Listeria only differed by 1 to 8 SNPs. The food isolate from 88 and the human isolate from 2000 differed by a single SNP. That Listeria basically stayed the same in that processing facility over 12 years. So it shows that we can have Listeria with the same whole genome sequence survive in facilities over long, long times. It is not just Listeria. Here's an example of Salmonella, another classic outbreak, classic set of 2 outbreaks, 1998, Salmonella agona, toasted oats. 10 years later, Salmonella agona, unsweetened puffed rice cereal, etc. Same processing plant, same company, same Salmonella, survived in a facility over 10 years. So now let's move to case studies. But before I go there, any questions? Any question about what we sort of know about Listeria salmonella ecology in processing facilities, but also how does that extend to kitchens, retail establishments, or down to the farm?
And almost everyone is muted.
And so if you want to ask a question, you check, you might be muted. So you're either going to have you have a bar or a navigation bar either on top or bottom, and you can unmute yourself. Or you can type a question into the chat if you have one. Now we better gonna get ready to ask some questions because we're gonna get stuck on case study 1. There's nowhere to go if we don't get audience participation. All right, let's move to case study 1. So this is, somewhat hypothetical, but highly informed on real events. And once we've gone through it, I can explain what the real events were. So, all right, we have a couple of questions. Let's going to get one. One of them, have you found the same persistence of Listeria types in kitchens and food safety establishments? Food service establishments. So we have found Listeria surviving for up to at least 12 months in retail delis, in supermarkets. In one occasion, we actually was traced back, and this was work we did with New York State Ag and Markets, very specific to a retail deli case that was basically uncleanable and sanitizable. In kitchens, there have actually been a few outbreaks, and I think New York City Department of Health might be able to comment on them, where it appears that Listeria survived in a hospital kitchen over time, potentially in a slicer or some other equipment. Similar things have been found in Europe. There have been a number of outbreaks, a couple of outbreaks in the UK of Listeria of survival in kitchens and in hospital kitchens. And there's also been a case where Salmonella has survived in a slicer over time in a retail establishment. Home kitchens, very few people have done the studies needed to do that, but I think it's very, very safe to assume that we will find persistence in also food service establishments. The question is whether we're going to get enough exposure to get a real outbreak. Okay, so here's case study 1. We find Listeria monocytogenes in the floor drain of a large hot dog processing facility. Big facility, makes 50, 100,000 packages a day. We find it in the floor drain of that facility. It is the year 2017, so USDA wants to have this isolate as one of their routine swabbing events that they do in these facilities. They perform whole genome sequencing. Whole genome sequences shows a match to 2 human isolates where both people, as part of the routine Listeria questionnaire, so every case patient with Listeria, as far as I know, is going to get a questionnaire administered. Both said they ate hot dogs, provided different brands when re-interviewed, and both of these brands are actually produced at that given facility. The 2 isolates, so human 1 and human 2, human 1 differs by 2 SNPs from the floor drain isolate. Human 2 differs by 5 SNPs from the floor drain isolates. And let's say, for argument's sake, this facility and the 2 cases are in the same state, the same jurisdiction, and any one of you on this session today now is like confronted with, say, okay, guys, this is what we found. Yeah, this happened, a couple of months ago. What are we going to do? What are the next steps? Where are we going to go with these sort of data? And so this is now where we're going to ask for audience participation. I'm pretty sure Andy is on, Madhu is on, I think David is on. So we have a couple of people who can definitely jump in just to sort of get us started on the conversation here. So here's my plea for everyone. Someone tell me, what would happen in your jurisdiction? What do you think people would do if they're confronted with this scenario? Okay, we're going to unmute everyone just so, to lower the entry barrier to a comment here. Yeah, if you prefer.
Okay, this is Andy. I'll jump in.
Okay.
Given the serious nature of listeria, I would work with our environmental health folks to maybe start a review at the facility, especially given that it's listeria.
All right, so you would send people to the facility. So what would you ask them to do when you say a review of the facility? What do you think should happen in that facility?
I'd ask David. No, but certainly review past inspections, have them review cleaning and sanitation processes, and also seeing if that floor drain is the same, as it was before, figuring out if there could be ongoing contamination there, and also looking for other points of potential hard to clean surfaces.
Anyone else? David, what would you instruct your people to do in there? What would you do if you go into that facility?
So basically, similar to what Andy said, but we would definitely want them to do an environmental assessment at the facility. And one thing that jumps in mind is, the 2 human cases that eaten hot dogs with 2 different brands, both produced in this facility. I'm wondering if they were produced at different years, and if it's example from 1998 to 2008. Do some environmental sampling of same equipment that may have been around at both of those times, or basically the locations of processing, if they were in the same areas both of those times for these products, to focus on those things in common there. But definitely do an environmental assessment and do some sampling.
Okay, so we would go out, do an assessment, but also collect some sponge samples from the environment in the hope of finding some more Listeria monocytogenes, which we then, of course, could also sequence and see if that same Listeria monocytogenes subtype is still in that facility. Let's say we find it, and we find it again in a floor drain, but it's same proximity, but that floor drain is 50 yards away from any exposed hot dogs, but clearly that subtype is there. Now that we find it, it's got a few more SNP differences, but it's basically the same. So what will we do next? Ask USDA to initiate a recall? See whether we can talk the plant into a recall? What do you think is going to happen here? Is that sufficient evidence? What does anyone else think here? What's the alternative scenario? So what else could explain this finding here?
Well, this is David again. I mean, we would definitely try to push for USDA to do something and hopefully maybe do a recall, or if not, put out an advisory about this hot dog brand, since we've got human cases, so close related. We, want to prevent deaths from occurring and try and prevent this product from causing deaths. But in regards to the facility and that floor drain, I'm wondering what type of equipment may have gone across there and brought into the processing facility where the hot dogs are made and going across that floor drain. I mean, we've had issues where we found Listeria on carts that were transported from one area to another.
Who would request that we're going to try to find these brands, find some what we call library samples? So sometimes these facilities have samples from over shelf life. If these hot dogs have a 60-day shelf life, they might have some older samples. Would anyone say, let's go full out and get 50 to 100 packages of these hot dogs and sample these actual hot dogs to see whether we can find an actual product rather than just a floor drain? What else could one do to sort of, so it could be this facility, right? But could it be that multiple facilities produce the same brands? Could you have the same subtype in a different facility potentially? So, anyone else here? So what I would probably push very strongly for some finished product sampling. At least identify whether other facilities, same company, but other processing plants make the same brands. I'd do some investigations in those too. And we'll show you some data and say it's not always for sure that you're going to have subtypes unique to a facility. Listeria could move from one processing plant to another. It could be in different processing plants. So just finding it a drain, you're probably going to get, USDA will get considerable pushback from that facility. With 2 cases, can you do a case control study and get something significant? Show that association with hot dog is significant? Probably mathematically, by and large, impossible. You don't have enough sample numbers, right? So with these few cases, you're starting to push the statistical boundaries of our sort of traditional tools, such as case control studies. So it could be pretty challenging. I think if we don't identify that Listeria or a Listeria in a finished product, then, or find some other evidence of unhygienic conditions in this facility, we may or may not be able to shut down this facility, and we may or may not want to shut down that facility because maybe that facility isn't the source, maybe it's another facility. We shut down one that produces safe product that then shifts the whole supply to another facility. That might now produce more, and that might be the actual source, and we might actually increase an issue rather than control an issue. So let's go to another case study and then gonna go back to some data that support what I'm talking about here. So very, very similar, but now we have Listeria isolate from a deli case in retail. So agent markets in one of the states on this call went in. For some reason or another, decided to take some sponges in a retail environment, in a deli case, find Listeria monocytogenes, whole genome sequence it. That whole genome sequence matches 1 human case. So rather than 2, now we got 1 human case, 5 SNPs. That patient died, but a relative recalls that she did shop at the same chain. Not sure whether she shopped at the same store where the deli case isolate was obtained. So, single case, some what I'm going to consider weak epi, potential epi. What are we going to do? What does this mean? Does that mean that a retail deli is responsible for that case?
This is Madhu. I would probably want to take a look and see if the chain had a loyalty card and try to get a shopper card history from the deceased case, if possible. And then also if we knew specifically what they purchased from the deli case.
So that will be one. Definitely see whether we can create some stronger epi here. Anyone else? What else would we do? So I'm going to now just give you some real-world examples that sort of illustrates the challenge in this. And this is actually a paper that we published more than 2 years ago, where we actually did whole genome sequencing of Listeria monocytogenes isolates from more than 15 retail deli establishments that were located across different states: New York, Indiana, and North Carolina. In a number of cases, we found basically identical isolates, let's say 0 to 3 SNP differences across different retail delis. In one case, the most intriguing one, we found, and I'm going to call them identical for just simplicity's sake, less than 3 SNP differences. We found identical Listeria monocytogenes in retail delis in 3 different states. Typo there, not there, but in 3 different states, retail delis had Listeria monocytogenes that could not be differentiated in any meaningful way by whole genome sequencing. In 2 of them, we only found that specific subtype once. In 1 of those retail delis, we found that subtype over time. So multiple visits. So each of these delis were visited between at least 6 to 9 times. Found the same Listeria monocytogenes multiple times. What do you think happened there? How could you find the same L. mono in retail delis in 3 different states? Same suppliers, one answer, and that's what made my suspicion. We have a supplier, we have a large processor that makes deli meats. They have a contamination issue. Those products are delivered to retail delis across different states. We re-pick them up because from the product, they contaminate the environment. We pick them up in the environment sporadically in 2 of these delis, but in 1 deli, it was introduced into the environment from a supplier, found a niche, found one of these hiding places that I've shown you. Whether it's a slicer that isn't being cleaned perfectly, not going to be disassembled enough, whether it's a deli case, whether it's a floor drain, God knows what, and survives there over time. Now we have a single case, we might pick it up just by chance in 1 retail deli, but in reality, that same Listeria might have been in that 1 retail deli just for a day or 2. It might persist in another retail deli, and the real root cause would be what I would call upstream. That's at a supplier, a distribution center potentially, even though not very likely in the case of Listeria, but potentially possible in other cases, etc. So sometimes these subtype matches with whole genome sequences don't get us to the real root cause, but have sort of these accidental findings, and we can pick up the same Listeria monocytogenes in very, very different environments, particularly with these complex supply chains. So as we use whole genome sequencing, really understanding the realities of supply chains, they can be very, very different for different products and can change very quickly, is very important. And so this one illustrates with some real world data that we can indeed find the same whole genome sequence of Listeria in different environments. So this sort of is obviously relevant for both of the case study 1 and case study 2. It also would not be, while I've not seen any clear evidence, it would not be impossible to find the same Listeria monocytogenes into processing facilities, whether it's introduction from raw material, introduction from ingredients, maybe even introduction through people that move between facilities. This has never been shown or proven, but it is, and while it is potentially unlikely, it is not impossible. And at some point, we might pick up some cases like this as we do more whole genome sequencing. And sometimes it may become the epidemiologists and the epidemiologists at state or county level that don't really have to drive these investigations to find some of those links that we have not thought about previously. Okay. Let's move to another organism. So let's take case study 3. So Salmonella isolate is obtained from goose feces at a university research farm. And this sounds sort of goofy, but I'll show you that again, this is actually based on real events. In a certain state, as whole genome sequenced, maybe be part of a research project, added to the database. And at some point, it turns out that we have 3 human cases that matched that Salmonella from these goose feces that was picked up in this research farm. And this is a research produce farm. And they're very similar, these 3 human cases, to this Salmonella isolate. 2 of these 3 patients actually report consumption of tomatoes, and the research farm is located in an area that is known for tomato production. We could call it the Delmarva Peninsula, whatever we want to, okay? So, next steps. Does that prove that tomatoes from this area are responsible for these 3 cases? Or the other explanations? What do we need to do here? Let's start with that. Are there other alternative explanations that could explain this other than must be tomatoes from this area that were contaminated with salmonella and caused these cases? See, there are a few people here who haven't spoken up, so someone want to give it a shot here? What else could explain this? Oops. Could be that these people worked on a farm. Good idea. So it could be with salmonella. Of course, we often think about, our first thought might be foodborne exposure, but with salmonella, we can have other routes of exposure. So zoonotic transmission, could be working on a farm. Another comment was, could the people come in contact with goose feces at a park? Absolutely. The other thing, so let me give you some more background why, again, to convince you that this is not purely hypothetical. So this is a paper that was actually published, and I took a small part of it, but it talks about the strain with the smallest number of SNP difference to the clinical isolates in clade A are 2 isolates from ARAC. And ARAC is an abbreviation for University Research Center, actually. One from goose feces, isolated September 2010, and one from creek sediment, isolated in August of the same year. So in this case, you can see these are co-authors from FDA, and they found this, at the very least, fairly intriguing. Now, one thing I did is, just for argument's sake, took a look at sort of flyaway patterns. So obviously, we can see that birds and Canada geese, while some of them are stationary, some of them migrate, have different migration routes. Is it possible that a bird, whatever your favorite bird is, comes all the way from the south and like this fly route like this, or come and deposited feces somewhere here, somewhere here, somewhere here, and even moving into Canada? Would we find, would the whole genome sequences of that salmonella deposited along this flyway be different? Or would we find the same whole genome sequence of salmonella in different locations along bird migration routes, potentially? Don't think anyone's done that research. It's non-trivial, but again, we can't necessarily exclude that. So the point is, there are some very effective means of potentially moving salmonella, listeria, and other foodborne pathogens with a given set of subtypes over decent-sized distances very quickly. One of them, obviously, is supply chains. I have a processing facility in the middle of the US, and it distributes all the way from the East Coast to the West Coast. Can very quickly transfer listeria, salmonella with the same subtype that way. Another way is some highly mobile wildlife, in addition to highly mobile humans, but highly mobile wildlife, obviously. Birds come to mind as some of the most mobile wildlife. So I could, in this case, say, well, could be, but let's also figure out, what are these geese? Let's find out a little bit more about these animals. Let's look at some of their patterns of movement. Is there a possibility that this could just be an accidental finding, and that we could find similar whole genome sequences of salmonella in different areas? Maybe these birds even came from Mexico, from South of Latin America in general, and we could effectively find similar subtypes in different parts of the world, different countries, not just different regions of the US. So again, we now need to move very quickly. Maybe it's not just about epidemiology. We need to understand pathogen ecology, wildlife ecology, to try to at least identify how plausible is it. And the challenge, as you can see from most of these studies, is that we deal with very few cases. So even with 2 cases, 3 cases, very hard to get reliable epidemiology. If 2 cases ate tomatoes and one of them remembers that brand of tomato and they came from the region in question, that doesn't prove that that's the source for both of them. So we're pushing the boundaries and limits of epidemiology with some of these rare, as we move into outbreaks that are defined by 2 or 3 cases potentially. Yeah. Now let's move to another commodity. So you see we move pathogens and commodities. Now we think about a larger outbreak. We have salmonella isolate from a raw chicken processed in facility X, subjected to whole genome sequencing. And in this case, we find 150 human isolates that differ by 1 to 5 alleles from this chicken isolate. So this could be in our year 2018, 2019. We do routine whole genome sequencing of salmonella from human cases. A finding like that would potentially be feasible. Of these 150 patients, only 19 people recall brand names. These people typically consumed, 135 of these 150 patients consumed rotisserie chicken, similarly prepared chicken. 19 people sort of recall some brand names for whatever reason because they bought the chicken and made it themselves. 11 of these, the brand names that come from these people represent brands actually produced by that facility. So we have a big chicken slaughterhouse linked to 150 human cases. But at the end of the day, it only boils down to 11 patients that represent that brand. What does that mean? Does that mean that should we shut down the chicken slaughterhouse? They're responsible. You could make it more complicated or more challenging by saying these salmonella are multi-drug resistant. There's only one remaining antimicrobial that really works on this salmonella, so bigger public health hazard than even your average middle-of-the-road salmonella. What are you going to do? What does this mean? Are there alternative explanations for a finding like that? Are there explanations where shutting down that facility might not necessarily be in the best interest of even public health? What do you think? So first thing is, where could the salmonella have come from? What are some alternative explanations for these findings other than, the most obvious one is there's a problem in this chicken slaughterhouse. Andy, any thoughts on this one? I'm going to have to come back to you and David since. Just going to give people time to think about this. You can discuss it with your neighbor if you have one in front of your computer. What do we think are some other possible explanations for this? Some things that we want to consider. What type of investigation will we launch? Where will we look? So on the epi end, so one of them was, I want to know where they bought the chickens. Very good point. So we want to see is there a common source maybe downstream. So these ones happen. This is a widespread outbreak. So this is an outbreak that didn't just happen in one state or one legislation. This is across 15, 20 states at least, all the way from the East Coast to the West Coast. And that chicken processor is a national brand. It's actually a very predominant brand. That processing, that brand, that company owns about 30% of that. Produces about 30% of the raw chicken that is sort of pushed out throughout the US. Another question is, what about other facilities that produce the same brands? What is their testing showing? So that there is no testing result, but maybe there are other slaughterhouses that produce the same brands. And the question is probably should send some investigative teams into some of the other facilities and see whether we find the same subtype in another slaughterhouse that's in a different state, maybe even that produces for the same brand. Another question was, what may be the on-farm prevalence for this salmonella in chicken? We don't know. We know that it's a common serotype that's found in salmonella. For argument's sake, in chickens, let's say it's enteritidis. But we haven't really done that many studies. There aren't that many salmonella isolates. Actually, it turns out there are virtually no salmonella isolates from chicken farms in our whole genome sequence database. So difference between this case study and the other ones is obviously now we're talking about a raw product, right? So could it be that that salmonella came from a chicken farm? a chicken farm? Could it come from the farm that raises the parents of the chickens, that makes the eggs, that makes the broilers? Chicken supply chain is actually pretty complicated. There are a few breeders that breed some of those chickens where then the parents come from that breeder, and then the parents make the eggs, and the eggs go to these broiler farms, and then these broilers go to the slaughterhouses. So this source could actually be very upstream. A worst case scenario that I would worry about it, again, this is an accidental finding. Facility X has that Salmonella whole genome sequence type on their chickens, but it's only 1% of their chickens that are contaminated with it, and it's typically low levels. Why? Because it's a very advanced facility. That facility happens to do air cooling, very good sanitation practices, very good practices overall, some interventions that reduce Salmonella numbers, a very good facility. We just happened to pick up one chicken that had it. There's another facility, Facility Y, that we did not investigate. That one has 10% of their chickens positive as the Salmonella. And the Salmonella actually is coming in from upstream because it's really living and persisting in a parent flock. Now, by us shutting down Facility X, we actually potentially might shut down a facility that makes the safer chicken and move and shift the supply chain to another facility that actually may have a higher level of contamination because the contamination source is way upstream. So again, an understanding of supply chain, bringing in people who understand those supply chains, and then investigating upstream may be important because the actual source, the actual root cause may be way upstream in that overall supply chain. So just some considerations along the lines of what we covered. And I'm going to be honest, I don't even pretend to understand the full complexity of poultry supply chains, particularly as we move to broilers, but similar to eggs and egg-laying chickens. So I see quite a need for better interactions between, in this case, obviously USDA, epidemiologists, as we move into whole genome sequencing of Salmonella, and going to start to see some of these potential matches between human cases, a few or many, and isolates that come from foods, raw foods, processing plant environments, etc. I'm going to finish with a final case study that actually moves us more into an area that may not be directly relevant to some of you on the call, but again, is informed by real action, is something that already happens, but does not involve human cases. So in this case, we have a processing plant that makes a variety of different meat products, ready-to-eat meat products. Salami from a processing facility, this processing facility is found positive for Listeria monocytogenes, and then actually a closed package was picked up in a supermarket. Listeria monocytogenes whole genome sequence, no match to any human isolates, all human isolates at least 30 SNPs away. What action would that company that makes this salami, so plant A, because their plant code is on that package, have to take? Not a trick question. Someone want to humor me with an answer here? What would have to happen for that? What does plant A have to do as an action based on this finding? Recall, exactly. Recall, what would the facility have to recall? Most likely just that one specific lot, salami produced with that lot date on that date. Even if there are other products produced that same date, maybe prosciutto, some other product, depends on the root cause analysis. Obviously do an investigation. USDA may do another investigation. If they find nothing else there, no other Listeria findings, it could be a very limited recall of one lot, one day. Now, let's take this forward. It is 11 months later. Another sample from the same facility tests positive for Listeria monocytogenes test by regional government labs, state ag markets, for argument's sake. Again, it's a packaged product. It's in its original packaging, not exposed to the environment. Again, whole genome sequencing done, no match to any of the any human isolates in the database, but this isolate differs by the isolate from January 2016 by only 5 alleles, or 5 SNPs, if that's how you want to think of it. So very, very similar isolate found 11 months later in a package. What action does the processing facility have to take now? What would USDA, because the USDA regulated facility, how would they approach it? What would they be concerned? Anyone want to take a stab at that? So on the one extreme, it could be another one-day recall, right? But what might these data suggest about this listeria? Well, based on some of the things that I've shown you, possible that this is persistent contamination. This listeria survived in this processing facility over 11 months. It survived in this processing facility for over 11 months. One could conclude that there's a high risk that any of the products produced between January 2016 and December 2016 are contaminated. One could conclude that that facility was running under unhygienic conditions. Food cannot be produced under unhygienic conditions. So this could go down the route where we require an environmental assessment, where we may have to do some more testing, but even in absence of any further testing, it might be what we call a bracketed recall. That facility may have to recall everything produced there because the public health risk is, even though public health risk doesn't seem to be that high because we don't have any human cases, but the production practices were such that this is not a food that was produced under the types of hygienic conditions, and that processing facility did not have the environment under control, so there might be other issues that are not recognized. So these are also the sort of things that have happened. There have been recalls like this with PFGE, 2 or 3 isolates from the same facility over 15 months, same PFGE type, no human cases. Environmental investigation identified some issues, maybe imperfect sanitation, this and that. Recall of all products produced over 12 to 15 months, plant shut down for months until they could clearly show that they identified the root cause of the problem before they could restart. If you want to, the story is more or less in the public domain. It's a company called Boreas Foods that had to shut down one of their facilities years and years ago based on repeat PFGE positives with the same subtype. We've likely to see some more of that with whole genome sequencing. At some point, this may or may not involve public health epidemiologists, but it could, and I expect that at least some of you on this call are going to get roped into investigations like this. So with that, here's some conclusions I take from this. I think the importance of understanding that foodborne pathogens with close related whole genome sequences can be found in very different locations. We talked about distribution through supply chains, whether that's one processing plant to many retail operations, whether that's a parent flock of chickens or a breeder flock where these birds, these live birds, get distributed to many different farms, and then from these different farms to different slaughterhouses or egg processing plants, or whether it's birds or other wildlife that moves fast, and then people that move between facilities potentially can also not be included. Second conclusion, that when we start to see more and more characterization of pathogen isolates from foods and food-associated environments, we will find more potential, maybe real, links to human cases. Whole genome sequencing alone, because of what I said in bullet 1, is not sufficient to say, same whole genome sequence in a food or processing plant in a human means cause and effect type relationship. Epi investigations are needed. They can be challenging because very often we may only have a few human cases. Epi follow-up needs to consider the complete supply chain, microbial ecology, and alternative hypotheses, and it's going to be called upon more often to investigate those. At the end, This may lead us to a need for new, different data analysis approaches that integrate epi and whole genome sequence data, or in general, just different approaches how we can deal with sort of epi of small numbers of patients when we have 2 to 3 patients. So I see a number of very, very interesting challenges that have tremendous potential to improve public health, but also will challenge us potentially in ways that we have not been challenged before. So I hope this gave you some ideas, prepares us for the phase where these things happen. I think we are exactly at our time. If there are any final questions, feel free to speak up or send me a chat. We will share these slides on our webpage. We'll follow up, show you where these slides are. If anyone wants the slides and want to use them in some of their own training, etc., let me know. I'm happy to share them. And if there are any other questions or you run into a situation like that, feel free to contact us at the New York Integrated Food Safety Center of Excellence. Our job is to help with, interpretation of whole genome sequence data and foodborne disease outbreak investigations. And we have expertise ranging across the food supply chains, and the question where we don't, we can find it for you. Any final comments, questions from anyone? Okay, we have one comment from a chat. Probably says goodbye or something like that. Okay, that was just a thank you and goodbye. Okay, thank you all for attending. Hope you found it useful. Bye-bye.